Hello, everybody. Welcome to the October Cancer Genomics Cloud webinar. I'm so excited today to present our guest speaker to you, Dr. Despina Ciolas. And I'm going to introduce Dr. Ciolas, and then I'm going to do just a few little sort of messages about the CGC, and then we're going to dive right into the presentation. You should be able to ask questions in chat. You may also be able to raise your hand if you are able to use Teams features. So Dr. Ciolas comes to us from Weill Cornell Medicine. Dr. Ciolas is an assistant professor of medicine and a physician. And in addition to treating patients, she conducts research on cancer genes that influence the immune microenvironment using preclinical models of pancreatic cancer and colon cancer. She's authored numerous publications and has received a prestigious Mentored Clinical Scientist Research Career Development Award, KO8, congrats, from the National Institute of Health. And she is a member of the American Society for Clinical Oncology, the American Association for Cancer Research, and American Medical Association, and the Hellenic Medical Society. Dr. Ciolas comes to us from our friends at the the Pancreatic Cancer Action Network. And the CGC and the Pancreatic Cancer Action Network have some important synergies that I want to highlight. Access to the TCGA and CPTAC is enabled for the PanCan Spark platform through the Cancer Genomics Cloud. So that allows researchers who are working within PanCan Spark to be able to compare those public data sets with the data from the PanCan sponsored studies. That lets you access molecular data and imaging data, but doing it in the comfort of the cloud where you don't have to download those files and then re-upload them somewhere. You can simply stream them to your analysis. So one of the things that we recommend doing if you're interested in doing this research is using our built-in data browser to actually filter files and add them to your cloud workspace. And CGC users will know that from there, you can find and select a lot of pre-built tools and workflows, create your own workflows. And we also have tools for tertiary data analysis using R, Python, SAS, or other different environments. So in general, what this would look like is going from data browser, finding the files of interest, bringing them to the CGC, and then running your analyses and using Data Studio for your downstream analyses. So that's just sort of really kind of a word from our sponsor, the CGC. And now I'm going to turn things over to Dr. Ciolas, and we're going to hear about this awesome immunotherapy research.
Thank you. I am so excited to be here today, and I'm so thankful for you guys having me here on a Wednesday afternoon. Today, I'm going to talk about my research on divergent immune responses in pancreatic cancer that are driven by tumor genotype. So, as an introduction, hello. I am an MD PhD, so that means I did my medical degree at SUNY Stony Brook in New York, and I did my PhD in genetics at Cold Spring Harbor. From there, I subspecialized into internal medicine and medical oncology. So right now, I am an assistant professor at Cornell Medical College, where I see patients with gastrointestinal cancers 1 day a week. And then I have my research lab here, the website's here, where we sit and we do research on gastrointestinal cancers. So I am a wet lab, the wettest of the wet lab biologist, but as a new laboratory principal investigator, I do have, as a new head PI, I have the opportunity to design my research lab in a way where we can use human clinical data, human data sets to identify scientific hypotheses and run back to the lab and test them out and try to uncover mechanisms. And this has resulted in a number of interesting projects. And so but today I wanted to talk about pancreas cancer. So the outline of my talk is to do an intro on pancreas cancer, and then talk about one of my previous project that has been published on P53 and the immune microenvironment, and then focus on what we're doing right now on KRAS and immunity. And pancreatic cancer is really at an exciting turning point, because we have access now to it, and I'm going to talk about this a little bit later, to genomic data. And so we have the ability to ask really specific questions that allow us to perhaps personalize specific treatment for patients. So building on that, as I mentioned, one of the diseases I treat are patients with pancreatic cancer. And this is difficult due to our limited therapeutic options and poor outcomes. The 5-year survival rate, although better than it has been in the past, is still 12.8% for all comers. And the incidence of pancreatic cancer is expected to rise. And so as it's expected to also then become the second cause of cancer-related deaths. And so why is pancreatic cancer such a difficult disease to diagnose and treat? And there are a number of different reasons. Some of them are clinical, such as a lack of screening and early detection. But there's also biological reasons for that as well. And one of the hallmarks that has been identified is this immunosuppressive microenvironment. And it's a unique feature of the disease in that it can serve as a barrier to both immunotherapy and tumor growth. So what is the tumor microenvironment? Is basically the cells. It's a network of stroma, blood vessels, inflammatory cells that surround and interweave amongst the cancer cells. They can mask the tumor and lead to immune evasion. So there's an urgent need to increase our understanding of how tumors create this immunologic barrier that can, again, lead to therapy resistance for both immunotherapy and chemotherapy. So let's talk about how pancreatic cancer develops. Tumors evolve from preneoplastic lesions called pannins to frank adenocarcinoma. And during this time, changes can happen into the immune system, to the immune microenvironment as the tumor progresses. Mutations in the oncogene KRAS are an early driving genetic event present in pannins, the preneoplastic lesions, and present also in 93% of tumors. Now, emerging evidence also suggests that once you start having genetic alterations, this can affect the immune cells around. And so there has been previous work that showed that if you have this particular KRAS G12D mutation, it could enhance mobilization of immune cells to the tumor microenvironment. But building on this concept, the appearance of additional alterations may further alter the immune landscape. P53 is the second most common genetic alteration. And so we were kind of interested to see, well, what happens to the immune microenvironment as the tumor progresses from KRAS mutation to the addition of P53. Now, most alterations affecting P53 are actually missense mutations, which affect the DNA binding domain. And so because P53 is a transcription factor, it can actually lead to different downstream signaling. So it can result in gain of function activity, meaning that this mutated protein is neomorphic And so I was interested in seeing if mutant gain-of-function P53 can exert a specific phenotype to the tumor immune microenvironment. So when I entered fellowship, I was supposed to just test out different mouse models with immunotherapy because it was the hot, new, exciting drug and see what happens. But what I saw that I began to see there were changes in genotype. I'm sorry, changes in response based on genotype. So I wanted to explore this further. And as I mentioned, we're specifically talking about a gain of function, not a loss of function. So it's a different type of protein. And so while the immune effects were unknown, what I'm trying to say is the immune effects that were directly attributed to this gain of function protein weren't known. And the model we use, which is still the model I use for my current projects, is an orthotopic tumor model, where you grow your cancer cells in a dish that allows you to genetically modify them, whether it's through knockdowns, RNA interference, CRISPR-Cas, overexpressing things. And then you can take them and then transplant them through a little mouse surgery back into the mouse so that it's in its native immune environment in a mouse with a syngeneic intact immune system and allows the tumor to grow. So I set out to ask the question, what effect does the acquisition of gain-of-function mutant P53 have on the composition of intratumoral immune cells in pancreatic cancer? To do this, we used CRISPR-Cas gene editing to create a specific gene mutation in our KRAS mutated pancreatic cell line. So the goal was to create isogenic cells that we could manipulate in vitro and then orthotopically put back in the mouse for immune studies. And we focused on the R172H mutation because its human equivalent was the most frequent P53 mutation in pancreatic cancer, and it had well-established gain-of-function activity. So I created these two different cell lines, and we started to put them in the mouse. And what we found was that the tumors that had the mutant P53 had more neutrophils present than the tumors that didn't. And this is, I don't know if you can see my pointer, but in any case, this is the flow cytometry plot. And what's also interesting is even when you account for differences in tumor weight, still there was more neutrophils present. And we confirmed this by IHC as well. But at the time, we didn't know where we were looking for neutrophils. Is this even a robust phenotype? Are they even present in pancreatic cancer? And it had been published using RNA sequencing data from the Cancer Genome Atlas that a neutrophil gene signature is not only highly expressed in pancreatic cancer, but that pancreatic cancer ranks second on average amongst these other 15 other cancers. And it had been proposed that neutrophils could be either pro or anti-tumorigenic, depending on the specific cancer type and also on the stage of the tumor. But in the context that we're talking about in pancreatic cancer, neutrophils are consistent with a pro-tumorigenic role, because not only is neutrophil infiltration associated with poor prognosis in patients, but even having a high neutrophil count is a negative predictor of overall survival, as is finding neutrophil chemokines in the blood. And to confirm that this wasn't a result of loss of wild-type P53 function, we also created another cell line that had a P53 null, and we saw no significant differences in neutrophils when we orthotopically implanted them. So this again supported the hypothesis that this was present due to P53 gain-of-function activity. Now, looking at the other types of immune cells present, what we found when we performed an examination of the T cell compartment was that there were decreases in CD3 cells, CD8 cells, and Th1 cells, although there were no differences in CD4 cells. But the inverse relationship between neutrophil accumulation and T cell infiltration was consistent with prior published reports using pancreatic genetically engineered autochthonous mouse models, and was suggestive of the creation of a pro-tumorigenic immunosuppressive environment. And neutrophils in cancer can inhibit T cells through a number of different ways. There's been reports that they can inhibit them by degranulation, by inducing apoptosis. And there had been also a different report also exploring that neutrophils are basically a bad thing in terms of they contribute to metastasis formation as well. Now, we delve more into the mechanism of how this occurs, and this has been published, so you can look up our paper. But we also wanted to further explore this in humans. So we examined the human pancreatic cohort of the Cancer Genome Atlas for genes that were significantly enriched in tumors with mutant p53 in comparison to wild type p53. And so using gene ontology, we saw an enrichment of genes in neutrophil-related pathways in human p53 mutant tumors compared to p53 wild-type tumors. So because I want to focus more on the clinical implications and how this project leads to my current research, I'm going to skip over more of the mechanism and go more into, well, what can we do with this kind of info? And so immunotherapy can be curative in some types of cancers, but the currently available immunotherapy is not effective in pancreatic cancer, except for 1% of patients who happen to have this phenomenon called microsatellite instability. So because of the lack of response to current-based immunotherapy in pancreatic cancer, different combinations, both with chemo and with new agents, are actively being explored in clinical trials. And one combination that was been pioneered by a colleague of ours, Bob Vonderheide, was the combination of CD40 agonist antibody, which is an immunotherapy that aims to improve antigen-presenting cells. So we were interested in seeing if we depleted neutrophils and improved T cell function, would this combination work in our pancreas tumors? So to do this, so just to give you background, the idea behind this kind of chemo immunotherapy regimen is that the CD40 antibody is thought to activate antigen-presenting cells that can elicit an immune response from T cells using tumor antigens that were released by chemotherapy-mediated cell destruction. So being that neutrophils suppress T cell function and proliferation, if we wipe out neutrophils or deplete them, would this be able to augment the therapeutic efficacy of this regimen? And so to do this, we established tumors in our mouse models, and we started depleting them with neutrophil depletion antibodies. And we tracked their tumor size, and we gave them the chemotherapy immunotherapy compound, and we basically tracked them. And what we saw was that in the tumors that were depleted of neutrophils but treated with the same chemoimmunotherapy regimen, there was a greater reduction in tumor volume when combined with the CD40 immunotherapy chemotherapy combination than there was with just the combination alone or just the neutrophils. So, and then what was really exciting was when we looked inside the tumor to see what happened, we saw that there were more immune changes. So there were more T cells. So in the tumors that were depleted of neutrophils and treated with the CD40 chemo regimen, we found more CD3 T cells, CD4 cells, CD8 cells, Th1 cells, all consistent with a greater anti-tumor effect. So although this has been published, it kind of brings this idea of bringing together genetics and genomics with immunity and immunotherapy. And so it was interesting because there was another paper that had showed that a p53 deletion gives you a different immune microenvironment. So that was kind of interesting to see. And this type of concept is not necessarily new. It has been shown in kind of other cancers, but it suggests that perhaps patients should be stratified for certain clinical trials or certain therapies based either on their immune phenotypes or their tumor genotype in order to create a more precision therapy. So after finishing my postdoc, came to Cornell and I started my own lab. And I was really interested in exploring the relationship between different cancer drivers and the immune microenvironment, or if they have different mets and therapeutic response. And I really wanted to build a program where we could use not only our mouse models, but human data sets to directly test hypotheses and also understand biological mechanisms. And this has to be done by forming an active collaboration between physicians, scientists of all different backgrounds. Now, when we look at the genetic landscape of pancreatic cancer, and look, this figure, although it's a beautiful word plot depicting where the font size of each word corresponds to the frequency of the mutation, it's more than 10 years old. We've known this for quite some time, that this is a landscape. This is a tumor that's dominated by KRAS mutations followed by p53. But although we've had tumor sequencing for quite some time, tumor sequencing was not recommended for patients until certain clinical guidelines and bodies changed in 2019 and recommended that every patient that comes in who happens to have a locally advanced, like a stage 3 or a metastatic disease, receive tumor sequencing to identify all, unfortunately, very uncommon mutations, but are considered actionable, which means that there may be drugs approved for these mutations, and it may even be in another, it may be FDA approved, but for another cancer type, but still may be applied to these patients. So because of this, that means we could start offering tumor sequencing to our patients. But that doesn't necessarily mean that all patients are going to have it readily available, right? Especially if you're not at an institute that has, let's say, in-house sequencing, or if you're a patient where your doctor has to send it out to an outside company. And so, but it really kind of allows us having this type of data available and patients enrolled in it really allows us to be able to kind of understand more about the disease and thus create new hypotheses, new types of clinical trials, and so forth. And one of the things that has been that has come to attention is the fact that even though pancreatic cancer is dominated by KRAS mutations, it's not the same mutation. It's actually a couple different mutations. So the most common is the famous G12D mutation, which accounts for 40% of patients. However, there's also a G12V mutation and a G12R mutation. So, and what has also come to light is that even though these are single amino acid substitutions at codon 12, they can actually affect tumor initiation, progression, and signaling. Well, okay, so just throw an inhibitor at it and then everything will get better. Well, the FDA approved KRAS inhibitors only target the KRAS G12C mutation, C, which is present in only 1% of pancreas cancer, making it largely ineffective for most patients, 99% of patients. And as I mentioned, immunotherapy can be curative for some patients, but not effective in pancreatic cancer. So neither of these are really practical options for patients. But in this, we recently published September 9th in Cancer Cell in collaboration with the Chandwani Lab and Dow Lab and others, that we now have clinical studies where we can track progression and what are the clinical aspects of patients with different KRAS mutations. And one of the things that has come out is that the type of KRAS mutation a person has matters. It affects their prognosis. So this is a Kaplan-Meier survival curve. And it shows, it's based on patients from the COMPASS trial who had their pancreas resected. And it shows that patients with a KRAS G12R mutation live longer than patients with a KRAS G12D mutation, suggesting that these KRAS mutation-specific interactions may impact disease behavior. So we had a hypothesis. Given there's a difference in survival between these patients, maybe they have differences in their immune cell composition or function. It's a loose hypothesis, but maybe. So we wanted to know if there was specific immune cell properties associated with these tumors. And to do this, we decided to go forth by using mouse studies and human data. Now, as I mentioned, now we have a lot more tumor sequencing of human pancreas cancer patients. But we also now have developments in CRISPR-Cas gene editing, where we can go back to the lab, create mutations which we can model and study to test out hypotheses. So as we were kind of formulating this project, the Dow Lab, our collaborators here at Cornell, published this amazing paper where they made these different KRAS mouse models with different KRAS mutations through CRISPR-Cas gene editing. So we said, okay, well, what if we take our KRAS mutated cells, which have either a G12D mutation or so they're pancreas cells derived from a KRAS mutant mouse, and we grow them up in a dish, and we just want to know, are they have differences in terms of proliferation? And they don't, actually. And this was also published in that Cancer Cell paper that came out a month and a half ago, where we look more in terms of in vitro, what's going on. It's between the G12D and G12R cell line, and there aren't differences in proliferation. So it's not simply a matter of one cell line being slower growing than another. But if we took our KRAS P53 mutant cell lines, we put them orthotopically into the pancreas of a mouse, and then we wait to see if tumors form, what we found was that the G12D tumors are more aggressive, they grow faster, they're bigger tumors than the G12R tumors. And interestingly, what we also found is that the overall survival patterns mimic what we saw in humans, in that the G12D tumor-bearing mice die faster than the G12R tumor-bearing mice, indicating that something systemic, something that requires, whether it's other cells like fibroblasts, immune cells, something that's outside that petri dish, happens once we put it inside a mouse. So this is really interesting. So number one, it gives us a nice model system that replicates our what we see in patients, and a way to study this mechanistically. But that doesn't mean there's an immune system involved. So we did look at inside the tumors through IF, and we saw that there were more T cells present in the G12R tumors. However, if we took our cell lines and put them in a mouse that lacks T cells, it was really interesting, because what we found, only T cells, technically also lack hair, but in this case, they have all the other cells present. They have B cells and K cells. But what we found was that the G12R tumors are now accelerated to the point where, if you look at them under the microscope, they look histologically like poorly differentiated tumors, and they die faster. Whereas the G12D tumors are not affected if you put them in a nude mouse, in a mouse lacking T cells. So that suggests that the presence of T cells are important, or rather there is an immune cell component that is important for G12R tumor development progression. So we wanted to explore this further. We also looked at dendritic cells. And at the time, what we found was that if you also put these cells in dendritic cell deficient mice, you get the same type of effects. And if you look inside those meaning that the G12R tumors are accelerated in mice that have lack dendritic cells. And if you look inside those G12R dendritic deficient tumors, what you find is that all the T cells go away. The CD3 cells and the CD8 cells, we're still quantifying the CD4 cells. So this was also very interesting, again, pointing to an immunogenic role for the G12R tumor. But is there immune data? This is a really cool mouse model, but is there immune data relevant to human clinical scenarios? And this is actually really, really, really tough, because what we're seeing is that we have this cool, interesting model, but we need specimens that are from untreated patients, because this model doesn't involve any kind of cancer treatment. And when looking for human specimens, a lot of times if a patient has a stage 4 cancer, rather than getting a biopsy of the pancreas, which involves an endoscopy, which is when, you requires anesthesia, they have to put a camera or with a tube down your esophagus. A lot of patients opt for a liver biopsy, which is done through the skin, although invasive, may not be as uncomfortable as an endoscopy. But we don't know how the liver may affect tumor immunity. And there's papers coming out mostly in colon and rectal cancer saying liver mets may affect tumor immunity, but that's not a topic for today. So we really want patient data where we need human tumors that come from the pancreas, not livers, and our pancreas, and we need to know if they're G12, I forgot. So we need to know if they're G12D or if they're G12R. And that's actually really hard to find, because a lot of times we would turn to different resources and they would say, we know they're KRAS mutated, but we don't know the type of KRAS mutation. You're like, how's that possible? And then another thing that occurred is they would say, yes, but we only have one G12R tumor and 4 G12Ds. And you're like, well, that's not really a big data set. So you needed like a robust data set that has these types of samples. So lucky for us, PDAC data was published from the human pancreatic cancer cohort. And it has been stratified for G12D and G12R, as well as other KRAS mutations, and shows that there is different downstream signaling between these mutations. And the other interesting thing is that this particular the pancreas cohort is 140 treatment naive, surgically resected, stage 1 through 3. Stage 1 through 3 means no liver mets, no lung mets. It's just pancreas, maybe lymph, and probably lymph nodes. Stage 1 through 3 pancreatic tumors that was collected for analysis. And so they also, the authors of this paper used a transcriptomics-based deconvolution method to delineate the cellular composition of the tumors. And what they found, and then there's an RNA-based immune cell inference tool called Excel, which was used to dissect the relative levels of different immune cells in these treatment-naive pancreas tumors. And what we found was that there are more dendritic cells present in G12R versus G12D patients. So we were like really excited, very, very excited. And so that's kind of that, because it validates our mouse model, but also gives us an idea that what we're seeing is clinically relevant. So we went, let me just take a second. We started off with the idea that clinically speaking, a G12R patient lives longer than a G12D patient, right? We don't know why. We went to a mouse model where we're seeing differences that mimic what we see in the patients. And then we went back to the patients to kind of tell us, is this true? And the presence of, and this is also, so while we're digging up all this info, what we discovered was that the presence of dendritic cells, CD4 cells, which I didn't talk about, but that's also clinically relevant and mechanistically relevant, and G12R genotype, all independently associated with prolonged survival in pancreatic cancer patients. However, a clear mechanistic connection between these phenomena has not been identified. So we're still on this hunt to try to figure out what's going on. And we collaborated with PanCan, who has a Know Your Tumor data set in Spark. And it's different because it's considered a real world data set. And they can always correct me if I'm wrong, but basically, this data set is available to research through a subset of patients who participated in the Know Your Tumor clinical program. So the Know Your Tumor clinical trial, which has been published, is to understand the unique differences between a patient's tumor. And so they would provide sequencing for patients to determine whether or not, just how kind of what I said in a couple slides ago, if you have one of those few uncommon mutations, but happens to have a drug, what we call actionable or targetable, then you would be able to provide that for these patients. So it's different in that it's a real world data set that can provide us with information. It's also publicly available through this website and has been published. So we got together with PanCan and we asked, so again, I'm asking a lot of things. I want to know, we were filtering through all of the patients in your database. We want to know which patients have available RNA-seq data. We want patients who have a tumor biopsy from the pancreas. No mets, right? Take away the mets. I don't want liver biopsies. I need pancreas. And we have to filter them for G12D and G12R mutations. And we have a sizable amount. So that was great. We were really super excited. And one of the things we did is we did an analysis of the top hallmark pathways enriched in cancer cells using MSigDB gene sets. And what we found was that G12R tumors have an enrichment in all these inflammatory pathways, interferon gamma, interferon alpha, et cetera, et cetera. And so that was pretty exciting. But I also have, in humans, but I also have some mouse data that, again, of our tumors that show those same signaling pathways, right? I didn't tell them that. I just said, let's do this analysis. And then I was like, guess what? So any case, again, pointing us to a potential mechanism for what we're seeing in the lab. So in summary of this part, G12D mouse tumors are more aggressive than G12R tumors, and our mouse model data replicate overall survival patterns in patients. G12R tumors manifest a distinct tumor microenvironment with increase in dendritic cells and T cells in comparison to G12D tumors. And analysis of publicly available and real-world human data sets also show an enrichment of dendritic cells and other immune-related pathways. So we're further exploring this. There's a lot of work to do. Everyone's working. We're working hard. But we're really excited about where this project is going and how it's coming together. So I need to thank the people in my lab who work really hard, our students, our technicians, our postdocs, our clinical residents, and so forth, and visiting fellows. I need to thank the collaborators, both the Chandwani Lab and the Dow Lab, who this project wouldn't be able to happen without their help. I want to thank PanCan also for their expertise and advice and help with moving this project along. Finally, these are our research fundings. We have a great new grant from the Cancer Research and Treatment Fund, as well as support from the Department of Defense, the National Pancreas Foundation, the NIH. This is our website if you want to learn more, if you want to join the lab, if you just want to help us out. And then this is my Twitter handle, in case you're really bored and want to look at my Twitter. Anyway, thank you very much.
Thank you so much, Dr. Ciolis. That was an incredible talk. I feel like I understand so much stuff. And probably as soon as we end this, I'll be like, oh no, I don't actually understand any of it. But that was just wonderful and really very, very inspiring and hopeful. We do have a question from the QA that I would like to voice for you. The first question is, you talked about malignant progression from pre-malignant lesions. Do you find, or is it found, that T53 and KRAS mutations are driving malignant progression? Is this reprogramming of the tumor microenvironment occurring early during tumor development?
Yes. So basically, what you're saying is correct in that once you get a KRAS mutation occurs, you start to get PANNs. And we know that simply having the KRAS mutation, at least the G12D one, because that's the most studied one, does start to allow changes in the tumor microenvironment. So, and that has been published by the Barsagi lab and the von der Heide lab. And is there reprogramming of T early? So yes, so early changes occur once you get KRAS, and then once you get P53, further changes happen. And that's what we were kind of bent on showing.
Awesome. And then a second question. If certain mutations, for example, KRAS G12R, induce an anti-tumorigenic immune environment, wouldn't that then lead to tumor cell death? Why would these mutations be selected in tumors then? Is there a subtle balance between tumorigenicity and immune response for these mutations? This sounds like a collaborator.
Oh, great. Oh, excellent. So I think that's really interesting. And the cells that we're using do have alterations in KRAS and P53. And ultimately, these patients get cancer, right? Even the mice die. The mice aren't, it's not like the mice are walking around for years. These mice are dying. So while there is some sort of I don't want to call it anti-tumorigenic, there is an immune effect. It is enough to the point where eventually it evades the immune system, a tumor forms and the mouse dies. So it's not so much anti-tumorigenic as it is, there is more of an inflammatory effect. And maybe it's something we can harness or harp on. Because these tumors aren't static, right? These things change over time as well, especially in a human, more so than in a mouse. So we're really asking very specific questions, and trying to apply them to a human situation.
Awesome. Thank you.
Thank you so much. I really enjoyed being here.
This was a great talk. Thank you so much. I will go ahead and release people if they want the last 10 minutes of their hour back. But if we have any other questions, please feel free to pop them into Q&A. But thank you so much, Dr. Ciolas, for being here. This was a wonderful talk.
Thank you, Sarah. I really enjoyed speaking to your group, and I'm excited about all the capabilities that we can do with your platform, the CGC platform.